Hello students, myself Pragya Ranjan. Going to teach you Unit V, Animal Physiology containing 18 marks, Chapter 17 Breathing and Exchange of Gases. On platform of PKP, let's start with breathing. It is the inflow, inspiration, and outflow, expiration, of air between atmosphere and the alveoli of the lungs. Respiration. It is an oxidation process of food substances within the cells to form CO2, water and consequent release of energy. Breathing. It is the inflow, inspiration, and outflow, expiration, of air between atmosphere and the alveoli of the lungs. Respiration. It is an oxidation process of food substances within the cells to form CO2, water and consequent release of energy. Respiratory organs in animals. Organism. Protozoans, sponges, coelentrates. Direct respiration, surrounding environment and plasma membrane by diffusion. Free living flatworms, general body surface by diffusion. Roundworms, no exchange of gases. Anaerobic respiration, earthworm. Cutaneous respiration, cockroach. The respiratory system includes spiracles, tracheae and tracheolas. Scorpion, spiders. Book. Lungs, fishes, gills. Respiratory system in humans. It comprises of nostrils, nasal chamber, pharynx, larynx, trachea, bronchi and bronchioles. Nostrils, holes of nose are called nostrils, external nurse, which leads to nasal chamber. 2. Nasal chamber. Nasal septum divides nasal cavity into two nasal chambers. The nasal chamber opens into the pharynx. Pharynx. It is a passage to both air and food. The pharynx leads to larynx. 4. Larynx, voice box. Larynx is a cartilaginous box which helps in sound production and hence called the sound box. During swallowing, glottis, a slit-like aperture in larynx, is closed by a thin elastic cartilaginous flap called epiglottis to prevent the entry of food into the larynx. The larynx leads to trachea. 5. Trachea. Trachea is a straight tube extending up to the mid-thoracic cavity. It is supported by C-shaped ring cartilage. When there is no air, trachea does not collapse due to cartilaginous rings. 6. Bronchi and bronchioles. The trachea branches into two primary bronchi, one to each lung. The primary bronchi are subdivided into secondary bronchi, tertiary bronchi, bronchioles, terminal bronchioles, and finally alveoli. Alveoli are the primary site where the exchange of O2 and CO2 between blood and atmospheric air takes place. Lungs. The branching network of bronchi, bronchioles and alveoli comprise the lungs. There is a pair of lungs. Lung has two membranes called pleurae or pleural membranes. The space between the two is known as pleural cavity and contains pleural fluid. Respiration involves the following steps: breathing, by which atmospheric air is drawn in and CO2-rich alveolar air is released out. Diffusion of gases, O2 and CO2, across alveolar membrane. Transport of gases by the blood. Diffusion of O2 and CO2 between blood and tissues. Utilization of O2 by the cells for catabolic reactions and resultant release of CO2. Mechanism of breathing. It involves I. Inspiration, E. Expiration. The main muscles of respiration in normal quiet breathing are diaphragm, intercostals, muscles of ribs, abdominal muscles. Inspiration. During inspiration, atmospheric air is drawn in. Total volume of air a person can inspire after a normal expiration. TV plus ERV equals IC. 500 plus 3100 milliliter equals 3600 milliliters. 2. Expiratory capacity. A. Total volume of air a person can expire after a normal inspiration. TV plus ERV equals 8. 500 plus 1200 milliliter equals 1700 milliliters. 3. Functional residual capacity, FRC, volume of air that will remain in the lungs after a normal expiration. RV plus ERV equals FRC. 1200 plus 1200 milliliter equals 2400 milliliters. 4. Vital capacity, VC. The maximum volume of air a person can breathe in after a forced expiration. TV plus ERV plus ERV equals VC. 500 plus 3100 plus 1200 milliliter equals 4800 milliliters. 4. Total lung capacity, TLC. Total volume of air accommodated in the lungs at the end of a forced inspiration. VC plus RV equals TLC. 4800 plus 1200 milliliter equals 6000 milliliters. Exchange of gases. Exchange of gases occurs in lungs and body tissues by diffusion. Partial pressure. Pressure contributed by an individual gas in a mixture of gases is called partial pressure. PO2 for O2. PCO2 for CO2. Exchange of gases in lungs. In lungs, the exchange of gases, O2 and CO2, between lung alveoli and pulmonary capillaries take place. The PO2 is higher in alveoli, 104 mm Hg, than the deoxygenated blood in the capillaries of pulmonary artery, 40 mm Hg. Therefore, O2 moves, diffuses, from alveoli to capillaries. On the other hand, PCO2 is higher in deoxygenated blood in the capillaries of pulmonary artery, 45 mm Hg, than the alveoli, 40 mm Hg. Therefore, CO2 moves, diffuses, from capillaries to alveoli. Blood becomes oxygenated, and moves to heart and then to tissues. Exchange of gases in tissues. In tissues, the exchange of gases, O2 and CO2, between tissue blood capillaries and tissue cells take place. The PO2 is higher in oxygenated blood in the capillaries, 95 mm Hg, than the body cells, 40 mm Hg. Therefore, O2 moves, diffuses, from capillaries to body cells. PCO2 is higher in the body cells, 45 mm Hg, than the blood in the capillaries, 40 mm Hg. Therefore, CO2 moves, diffuses, from body cells to capillaries. The blood becomes deoxygenated, and moves to heart and then to lungs. Transport of gases in blood. Blood is the medium of transport for O2 and CO2. Transport of O2 is dissolved gas. 3% of O2 is carried by blood plasma. 2. As oxyhemoglobin, 97% of O2 is carried by binding to hemoglobin, Hb. Hemoglobin consists of a protein called globin and a heme group. 4 iron atoms are present in heme group. Each ion has the capacity to bind to 1 O2. One hemoglobin molecule can carry 4 molecules of O2. 1100 milliliters of oxygenated blood can carry around 5 milliliters of O2. Oxygen hemoglobin dissociation curve. The percentage of hemoglobin IE bound with O2 is called percentage saturation of hemoglobin. The relationship between the PO2 and percentage saturation of hemoglobin is graphically presented by a curve called oxygen hemoglobin dissociation curve. The curve is sigmoid in shape. The oxygen hemoglobin dissociation curve is useful in studying the effect of factors like PCO2, H+ concentration, etc., on binding of O2 with hemoglobin. In the alveoli, high PO2, low PCO2, lesser H+ later on. Concentration and lower temperature makes the condition favorable for formation of oxyhemoglobin. In the tissues, low PO2, high PCO2, high H+ concentration and higher temperature makes the condition favorable for dissociation of oxygen from the oxyhemoglobin. Shifting of curve to the right indicates dissociation of O2 from hemoglobin. Shifting of curve to the left indicates association of O2 to hemoglobin. Bohr effect. Shifting of O2 hemoglobin dissociation curve to the right by increasing CO2 partial pressure. Increased CO2 concentration causing dissociation of O2 hemoglobin. Transport of CO2. 7% of CO2 in dissolved state in plasma. 23% of CO2 as carbamino hemoglobin. 70% of CO2 as bicarbonates. As dissolved state in plasma as carbamino hemoglobin. CO2 reacts directly with hemoglobin to form carbamino hemoglobin. As bicarbonates at the tissue site where partial pressure of CO2 is high, CO2 diffuses into blood, RBCs and plasma, and reacts with water to form carbonic acid. This reaction is very slow in blood plasma, but occurs very rapidly inside RBC because of the presence of carbonic anhydrase. Chloride shift. Most of the bicarbonates, HCO3, formed inside RBC diffuse out in the blood plasma along a concentration gradient. To balance ionic concentration, Cl diffuse from plasma into the RBCs. This shifting of chloride ions and bicarbonates between plasma and RBC is called chloride shift. Release of CO2 in alveoli. At the alveolar site where PCO2 is low, the reaction proceeds in the opposite direction leading to the formation of CO2 and H2O. Thus, CO2 trapped as bicarbonate at the tissue level and transported to the alveoli is released out as CO2. Haldane effect. Binding of O2 with hemoglobin displaces CO2 from blood. Bohr effects. Increased CO2 concentration causing dissociation of O2. Hemoglobin helps in release of O2 from arterial blood to the tissue cells. Haldane effects. Binding of O2 with hemoglobin displaces CO2 from blood helps in release of CO2 from blood to the alveoli of lungs. Regulation of respiration. Respiration is controlled by specialized center present in the brain called respiratory rhythm center. The respiratory center is composed of neurons present in medulla oblongata, dorsal respiratory center, and pons varolii, ventral respiratory center. The functions of the respiratory rhythm centers are again moderated by another center near pons called pneumotaxic center. Neural signal from this pneumotaxic center can reduce the duration of inspiration and thereby alter the respiratory rate. Chemosensitive areas situated adjacent to the rhythm center. Highly sensitive to CO2 and hydrogen ions. Increase in CO2 and hydrogen ions can activate this center. Activation sends signal to the respiratory center. Necessary adjustments in the respiratory process is made by which these substances can be eliminated. Expiration. Chemoreceptors associated with aortic arch and carotid artery also can recognize changes in CO2 and H+ concentration and send necessary signals to the rhythm center for remedial actions. Thank you to interact with us. Get subscribed to us for get firstly notified to you. You can also get me on Instagram. My id is pragya_ranjan_official. Okay, say you.